Voles are small, ground-dwelling rodents that are indigenous to Canada and the northern United States. They closely resemble hamsters in appearance and are related to lemmings.
In our experiment, we'll be examining the differences in mating behavior between two similar species, the prairie voles and the meadow voles.
Prairie voles are a monogamous species who form special bonds with one particular mate. Prairie vole couples mate and affiliate exclusively with each other, share nests, and even raise their offspring together. They form monogamous commitments called pair bonds, which is the ability to form intense, exclusive social attachments with a mate. Prairie voles have been of interest for researchers and to us because of their monogamous tendency being so similar to humans.
The meadow voles are a very similar species except for one key detail. They do not form mating pairs and instead mate with multiple partners. Additionally, they are known to exhibit slightly less aggressive behavior in defending their territory and mates. Scientists believe this is due to lower levels of two hormones, oxytocin and vasopressin, which promote bonding between male and female and more aggressive tendencies in males.
We chose to conduct this experiment in order to test the effects of hormones in mating behaviors.
Vasopressin, dopamine, and oxytocin, are they sufficient to bring about devotion?
Since prairie voles are monogamous due to the higher level of oxytocin receptors in females and the higher level of vasopressin in males, would injecting more of these hormones into the polygamous meadow voles cause them to behave more monogamously?
Our hypothesis is, if the increased hormone levels of oxytocin and vasopressin that cause monogamy in prairie voles were injected into the brains of polygamous meadow voles, then the meadow voles will behave more monogamously.
Conversely, the null hypothesis is, if the increased hormone levels of oxytocin and vasopressin that cause monogamy in prairie voles were injected into the brains of polygamous meadow voles, then the meadow voles will not behave more monogamously.
We predict that the meadow voles will become more monogamous from the prairie voles' hormones. Isolate 10 male meadow voles and 10 female meadow voles in a cage or makeshift habitat. Inject the vasopressin into the brains of the male meadow voles and levels of oxytocin into the brains of the female meadow voles. Record the tendencies of meadow voles to form pair bonds after mating, rather than mating with other voles of the opposite sex. Then observe the behavior of both the male and female meadow voles to see if they show signs of monogamous behavior.
Injecting hormones from prairie voles into meadow voles causes meadow voles to show more monogamous behavior.